we can probably get started with our first presentation. We have Stephanie Yelenik, who is a rangeland scientist for USGS, and she's going to be sharing with us today about some of the restoration work they've been doing, and her presentation title is Successful Planting of Metrosideros polymorpha, or OHIA, is possible in forest sites with active ceratocystis infections. And so with that, Stephanie, let me make you, I'm gonna make you co-host so you can share your screen. So when you're ready, Stephanie, you can share your screen and there you go, perfect. Thank you.
Does that look all right?
Yes, it does.
Okay. All right, hi everyone. My name is Stephanie Yelenik, and I have up until September of this past year worked with USGS, but I recently transitioned to US Forest Service, and I am also on the mainland at this point in time, which is why I'm in a sweater, because I'm always cold here. And I also want to point out my collaborators on this project, Kyle Roy, Jeff Stallman, and more recently we have the opportunity to add Corey Yanger, who many of you I'm sure know, to our projects. And while I'm on the mainland, she will be leading up the efforts with this rod restoration project, which we're super excited about. Okay. There we go. So rapid ohia death is caused by the fungal pathogens ceratocystis, huliohea, and leucoohea, and it can lead to really high mortality in forests. And as we all know, ohia is an ecologically and culturally important species. And just to go over some of the ecological importance of ohia, it is a canopy dominant species, and it actually makes up the bulk of the forest biomass in Hawaii. It creates habitat and food for our forest birds and other fauna, and it occurs over a really large range of temperature and rainfalls. It's our first lava colonizer, and as such, it builds soil and helps to capture water in our ecosystems. And so with the incidence of rapid ohia death moving across Hawaii Island and also now other islands, we need to start thinking about how we restore these important forests. And so as a restoration ecologist, when I think about restoration, I think about the different ways we can go about doing this. And the easiest and the least expensive way is to rely upon passive restoration. And so this is the case where, so here we have a picture from the Kohala where one side clearly has been cleared and ranched for a little bit, and then the other side is forest. And so it could be the case that if you remove the initial stressors that degraded the habitat, so you remove cattle and you stopped clearing any trees, and you fenced the area and you kind of crossed your fingers and hope for the best, that the forest would naturally come back. And so this is what we call passive restoration. And even passive restoration is quite expensive. I think as a lot of managers know, building and maintaining a fence is not a small endeavor, right? And so it's still an expensive endeavor. And in highly degraded sites, passive restoration is less likely to be successful. And then there's active restoration. So it's all the passive things that we just talked about, but it's also very actively planting in the species you want. And because this means that you have to hire people to collect the seed and grow the seed and have the greenhouse space and then plant them, prep sites and plant them into the ground, it's even more expensive than passive restoration. So we need to be careful before we embark upon active restoration to make sure that these activities are actually going to work. And there are very specific constraints to passive restoration in Hawaii. And today I'm going to be focusing on not just ceratocystis and rapid ohia death, but also invasive plants and animals that hinder passive restoration. So exotic animal species disturb vegetation, and more importantly, can disturb seedling regeneration. So even in a place where you might have native seed falling, if feral ungulates are constantly turning over that soil, they may not have the chance to germinate and grow. And also, in relation to rapid ohia death, feral ungulates such as pigs or cattle can wound ohia trees. And we now know that this is one of the major ways that rod can get into ohia trees and cause the death of those trees in our forests. And then there's exotic plant species in rod. So this hinders passive restoration in that native Hawaiian plants tend to have slower growth rates than many of the exotic species that have come into the islands. And because those exotic species tend to have fast growth rates, in the case that there is disturbance in a forest, whether that's from a hurricane or say a lot of canopy death due to rapid ohia death, it's more likely that those exotic species are going to be the ones that are successfully the next ones to come into that forest. So in this case, if we rely upon passive restoration, we're just going to be waiting for those exotic species to grow really fast. And so we've seen this in places with, for example, strawberry guava, where after disturbances, the species can really take over. And so in particular, because many of the forests that are being affected by rod tend to be in places with a lot of exotic species in the understory, this creates the condition where if those canopy gaps open up, the next thing to take advantage of that would likely be exotic species. And so because of these things, we're really interested in actively restoring these forests. So where is rod? And this is kind of an old map, but just the point is that it occurs all around Hawaii Island. And so from the Kohalas, which have really great native forests and native understory, and also, of course, in Puna, where you tend to have a lot of those invasive species in the understory that could be taking over those canopy gaps. And so when we first embarked upon this project, the reason why was because we heard a few times, like, why would I plant ohia actively? Why would I spend the money to do that when they're just probably going to die? And particularly in soils that have the rod fungus. And so we really just wanted to ask the very simple question, can you plant ohia seedlings in forests that are affected by rod and expect them to have any sort of survivorship? So the questions we wanted to ask with this research project were, can ceratocystis live in soils? If we plant ohia into these soils, how many will die due to ceratocystis? And then how does this compare to mortality from other factors? So in the beginning, I introduced these other constraints to restoration that we already have a lot of information about in Hawaii, and those are feral ungulates and exotic plant competition. So from a management standpoint, it really makes sense to try and understand which of these things are going to be the most important things for me to focus on with my management dollars. Like, where do I put my money, in a sense? So the first site in which we set up our experimental design was at KMR. So this is a military reserve, which is located next to the airport. This is lowland invaded forest with remnant ohia. And they had recently cleared road verges of invasives, and a lot of the ohia along those road verges were showing symptoms of ceratocystis, and some had already died. And so the first thing we did was tested these roadside ohia trees and the soils around them for ceratocystis. We then selected 10 trees that tested positive and 10 trees that tested negative for ceratocystis. And we planted ohia seedlings around those trees. So the adult trees are the big circles in the middle, and then we planted 6 keiki around each of these adult trees. And then to ask questions about competition from those invasive species and how that would affect those plantings, we ended up weeding half of the seedlings and then leaving the other half of the seedlings unweeded. So all of those bingabing and clydemia and all that kind of stuff could just grow up around those seedlings. Yeah, the experimental design was 10 each of those positive and negative adult trees, 6 seedlings around the tree, and we weeded half of them and we monitored monthly. And so this was really important because if you have, a tree that dies and then you leave it sitting in the sun for 6 months, you may or may not see ceratocystis if that's what killed that tree in the first place. And so we wanted to go in monthly and check all of our seedlings. And if any had died, we took them back to the lab and tested them for the presence of ceratocystis with both genetics techniques and then carrot baiting, which is just a way to try and foster the fungal growth. And then you essentially, it's like you take a sliver of the seedling and you put it between two carrots. It's actually really interesting. And then you just stare at it under a microscope for the little fungi. And so just to give an example of how fast those weeds can grow, you know, I have circled here an ohia seedling that we planted, and then within 3 months, it was completely covered by those invasive species in the understory. We also ended up fencing half of our plots because at KMR there are pigs that are roaming around, and we kept on losing our seedlings to those, and so we ended up fencing half of the plots. And so because we did that, and that way we can ask that question about, you know, what's more important to put your management dollars on? Is it worrying about feral ungulates? Is it worrying about the weeds? Or is it worrying about ceratocystis? And so to hop into the results, so we also, as I mentioned, we tested the soil around each of those trees, both the positive and the negative trees. So in around 2 of the 10 positive adult ohia trees, we found ceratocystis in those soils. And only 1 of those 2 had viable spores. And so here's that difference between finding the DNA and finding viable spores. The DNA in there means that it was in there at one point. The viable spores means that that fungus is alive and well and can attack something else. And around 1 of the 10 negative trees, it tested positive for ceratocystis. And so what this means to me is that those fungal spores are moving farther away from the foci. So like if there is a positive tree in a forest, that those spores are moving outward from there, and they're also in the soil around some of those trees that tested negative for ceratocystis. So there are viable spores of ceratocystis in the soil, and they can move around. That's kind of the take home from this. So this is the results after 1 year, and we have these published for those who are interested in restoration ecology. So we planted a total of 120 seedlings, and after 1 year, 41 of those had died. And the happy news is that all 41 of those trees were negative for ceratocystis. And so what this tells us is that, yes, planting ohia, like we always knew this, like some of your trees die in a restoration project, but within 1 year, they are not dying from ceratocystis, and then they're not succumbing to rapid ohia death. And so that's great news. And so just to break this down a little further, 71% of the dead ohia were in the not weeded plots, and 61% of the dead ohia were in the not fenced plots. And so from a statistics standpoint, what this means is that a seedling was 6 times more likely to die if you don't weed around it. So like this very simple task of weeding around your trees can really increase the survivorship of those seedlings. And a seedling was 3 times more likely to die if it was not fenced. So this suggests that fencing your trees will also increase survivorship. And so things that did not matter in this study were, was it next to a positive versus negative tree? It seems like, well, I mean, this isn't that surprising since they're not dying from ceratocystis in the first place. Whether or not it's next to a positive or negative tree also did not really matter. We also, when we were planting our trees, we were really careful to try not to wound them, because we know that's how ceratocystis gets into the trees in the first place. And so we took note if we had nicked the roots, or maybe a stem broke while we were putting the little tree into the ground, we would take note of that. But that also did not matter for whether or not those seedlings died. And so it's been some time, and we've actually been two years out in the KMR study. And so this is the 2021 update from this site. In total, 73% of our planted seedlings died. So yeah, there was a high mortality rate of those ohia. One thing I will say is that when we started this project, it was kind of a side project. It was, you know, it was a question that we were just chatting about at dinner one night with a bunch of friends. And we were like, hey, how come no one's just planting ohia out in these rot affected forests to see if they're even gonna live? And so someone offered me seedlings of ohia that were from a seed source in Volcano, and we planted them at sea level. And so what I'm saying here is that there is a distinct mismatch between where those seeds came from and where they ended up. And it might be that they are just not genetically how to say that they are not ready to cope with the hot, humid conditions down in Hilo, and they were more, well suited to the conditions up in volcanoes. So that could be part of explaining why so many of the seedlings died in the first place. 62% of the ones that died were not weeded, and 53% of the ones that died were not fenced. But just to put a finer point on this, in the end, so here's the difference between the fenced and not fenced. It's not that different, 41 dead versus 47, whether or not you fence. But when we look at the weeding, we see kind of a bigger difference. And I haven't run any statistical analysis on this, but it just kind of jumps out at me that the differences are bigger between who lives and who dies when you weed rather than when you fence. And so to me, what this is saying is that those competitive understory invasive species are really the ones that we need to be watching out for and dealing with in management when we think about if we're going to plant in ohia, or even what we have to deal with if we expect passive restoration of ohia. So conclusions from the KMR study were that successful restoration plantings of ohia are possible in forests that are affected by rapid ohia death. And that it's competition from exotic plants and to a lesser degree damaged by exotic feral ungulates that are more likely to kill seedlings than rod within the first year. And we also noted that bigger plants have better survival. And because rapid ohia death is occurring around the island, and the island has so many different ecotypes and climate types and substrates, we wanted to increase our study sites. And so we opportunistically started monitoring some restoration sites that other people had embarked upon. And so one of these is after the Keauhou fire, Hawaii Volcanoes National Park planted a lot of ohia into those burn sites. We've been monitoring 126 of those, 12 of which have died and none have tested positive for ceratocystis. We've also been working with the private landowner Upkaiviki in Hilo. We've been monitoring 80 of her planted seedlings. 10 of those have died. None of those have tested positive for ceratocystis. We also put in various sites where we replicated that same experimental design where we varied weeding and fencing so that we could ask that question across various sites. And so this is in Waiakea Forest Reserve.
I am sorry, Stephanie. I was trying to let someone in and I muted you on accident.
Okay.
I'm sorry.
Oh, no problem. So this is at Waiakea Forest Reserve on Steenback, and this is a site of a prior inoculation study, or where people actually specifically and on purpose inoculated adult ohia with ceratocystis to ask all kinds of biological questions that they learned a lot from. But we thought, hey, since we killed some trees, we'll plant some trees. And so we planted in a bunch of seedlings and did that same fencing, weeding kind of study. I don't have the full results for this, but as of this point, 8 of the ohia have died at Steenback and none have tested positive for ceratocystis. And then the final site is at Kalopa State Park. This is another site with active rod mortality. It has less invasive species in its understory perhaps than Steenback or KMR. And so we thought this would be an interesting contrast to ask questions about how important weeding is in a site with less invasive species in the understory to begin with. And so we have that same fencing and weeding study. To date, 26 of those ohia have died and none have tested positive for ceratocystis. So there is a story forming here, and it's a good news one, I think. So the implications from this is that ceratocystis can be viable in soils, and it's most probably from beetle frasts, looking at work that others have done. Planted ohia seedlings can live at least 1 year and even longer, even with infected soils. There is evidence, and we have seen this observationally, and other people are measuring this, that naturally recruiting seedlings will live in these same infected soils. So in spots where you don't have this massive invasive species understory, and maybe you have those nice nurse logs with moss that ohia really love to recruit in on, even if those forests are impacted by rod, I would predict that those seedlings would be fine, at least for the first couple years. But obviously this begs the question of like, we need to track these trees into the longer term. And so, 1 year, 2 years, that's great, but how long till these things are reproductively viable? They're creating their own seeds and making their own keiki. Like, are they still going to be okay at that point? So just from a management standpoint, passive restoration is still our least expensive option, and this could potentially work depending on whether or not we have those invasive species in the understory. So very well understood management tools, fencing, managing invasive plants, not wounding ohia. These are still our most effective tools at protecting forests and restoring forests from rapid ohia death. Planting ohia is certainly not a lost cause. And I would also offer up that we really need to better understand the importance of seed source. We have a lot of variability in the overall mortality of ohia across our studies, and we are not matching seed source to where we are planting. And part of this is just the logistics of collecting seed and growing them and waiting the 2 years when you have like your grant money in hand right now and you just need to find seedlings to plant. And so a real focused study on how important seed source is in restoration, I think is a next important step for forest restoration more generally in Hawaii. And so with that, I would just like to say mahalos to so many different organizations that helped us with this project and all the different people.
Thank you, Stephanie. That was really awesome. We're super excited to have you today. So if anyone has any questions, now is the time to put in the chat box and we can help to get those over to Stephanie. I guess the one question I did have, and I think you kind of touched bases on it at the end, was I was wondering, first of all, about seed source, like where you folks were getting your, your seedlings, and if you folks were taking a look at different species as you're, doing the restoration or planting. But
we are not, and one of the things we are looking at with Mark Hughes from Forest Service is the impact of rust. So we're also monitoring rust on these seedlings to see what impact that has. But as far as like our seedling source, we were just so scrambling to find ohia to plant in these different sites as we got the permits and the money to do it. And so at Kalopa, we sourced our seedlings from DLNR Nursery. At the Steinbeck site, we actually were given hundreds of trees by Lisa Keith's group that they didn't want anymore. And we ended up planting those in the field. That's probably the best match because they collect from often around Hilo is my understanding. And so planting up Steinbeck is pretty close to there. My hope is that the people at IPIF, like Christian and Susan, they are embarking upon their own kind of resistance project where they are very specifically planting in trees that were collected from that same site. And so there's the potential there to kind of ask questions about if you cross that, did you get differences in your survivorship as well?
Mm-hmm. Stephanie, I had a question, and I think you might have covered it a little bit, but I was just curious. Somebody had asked me, so I ask you, but if you eradicate an ohia tree that from your yard and you think it has its suspect rod, can you put another ohia tree in that spot, or would the rod be in the soil? What would you recommend for that?
Yeah, I mean, so I didn't specifically say it, but at Steinbeck, for example, every single tree that we tested around, the adult trees that we planted around, we tested the soils and they all had ceratocystis in them. They all had viable spores of ceratocystis in them. And to date, and I think it's, like I think we're 8 months in on that site, only 6 of like 138 trees have died or something. So, and none of them tested positive for ceratocystis. And so, I would say yes. I would say just go for it, especially if you have an affordable source of ohia trees. Like then I would say it's worth your while to try it out.
Okay, thank you. Thank you.
Stephanie, you mentioned that the size of seedlings can influence survivorship. Do you have a sense? I mean, if you had to give somebody a recommendation, obviously a a larger seedling or sapling is going to take more resources and be more costly. But do you have any recommendations for seedling sizes for outplanting?
I would say at least a 4-inch pot. Those were the ones that had the best survivorship at KMR. It's really hard to say because we source from so many different places, but you know, the ones that we got from DLNR were in dibble tube, like fairly large dibble tubes, and those have not been doing well at Kalapa. And those seem to be pretty root bound once, by the time we got them in, and that was partly us. We just, you know, there was just so much logistics in getting those plots set up and the permits and everything. So I would also say how root bound your plant is in those pots is really important. And I'm sure, I know that JB Friday would have a lot to say about this if he was here. But yeah, at least a 4-inch pot, and then not the kind of thing where if you're ohia, like if you're growing one on your own, and you know, like that kind of thing where the roots are just like coming out the sides, you might want to up pop that before you stick it in the ground.
Thanks.
Stephanie, I did have one question. Do you happen to know how long the viable spores persist in the soil? Someone wanted to know.
That's a really great question, and it's still, to my knowledge, an open question. I know that they've looked at it in logs. So like if they have an ohia tree that gets cut because it has ceratocystis, and then I know USDA, Lisa Kieslab, Mark Hughes, I believe, they did a study where they tested over time in that log, and it really kind of depends on the conditions it's in. So if you have your log sitting in a forest and it's moist and it's, you know, helo side, my understanding is that the ceratocystis will last much longer than if it's drying out in conicide or something like that. You know what I mean? Yeah.
Awesome.
And one final or another question that was posed in the chat box is if a person is interested in, this lady has just purchased some acres in Hawaii, Ocean View Estate, and she would like to do her part in perpetuating the regrowth of ohia. How would one go about doing that if they've got a large piece of land that they would like to use to propagate or grow more ohia?
I mean, it kind of depends on what that land looks like at this point. You know, if it's filled with invasive species and it's just going to take a lot of effort to clear and plant, you might want to just try out a few test plots, you know, so that you're not roaming around your whole property, and you're you know, just in the beginning kind of doing a little bit of gardening and figuring out for yourself, like how are the best way to go about doing all of these things. And I think just, you know, starting small also, you know, just for me, sometimes it's scary to jump right into a big project, right? And so starting small sometimes can just help us ease into those kinds of things and learn as we go. But, you know, first and foremost, clearing your invasive species around. If there are ohia on the property already, that's just the best thing to do. Also creating nurse logs if you can. So if you have trees that you fell, leaving those logs in place to kind of foster moss environments and little germination sites. Ohia really love to grow on those nurse logs, especially if they're above all of the invasive plants, or like there's grasses in the understory and stuff. That's another thing people can try.
Okay, that's a good start. Thank you.
I had a question, Stephanie, now that I know you folks, you have transitioned to Reno. I guess, what are the next steps for the project that's going to be that I guess Corey folks are taking over, or I guess, yeah.
Well, you know, since I left, unfortunately, they're going to be finishing out monitoring those various sites. And then, you know, we're closing out. And so USGS is going to eventually hire a restoration ecologist. So if there's any local scientists out there who are looking for a great job, I mean, there's so many different avenues of research that could happen. Or if you have ideas when that person comes on board, don't be scared to approach that person with your project ideas and the things that you think are important. Because what I can tell you, moving to a new region is I just don't even know what the open questions are. And so like I've spent my first few months here just going around to managers and being like, what do you want to know? What can I answer for you? And so have those ideas ready and don't be scared to talk to the new scientist that comes in, because you guys have the knowledge of like what's already gone on, right? And so you'll be able to help them direct their efforts. So unfortunately, I probably won't be involved moving forward.
Hey, Stephanie, it's Kim Rogers. I have a question. I'm curious. Most of the people that are here today are outreach specialists, so people working in conservation that we're hoping will share this information that's being shared today. And I'm wondering, I believe you've had a paper published on this. Is that correct?
Yes, we had a paper in 2020 in restoration ecology.
If you have that link handy, would you be able to pop it in the chat so everybody would have that at their fingertips, or the PDF if you want to upload the file? And then that's my question. I have a follow-up, of course.
Okay.
The follow-up is, what from your presentation today are you comfortable with us sharing? Or is there early stuff that you might have shared that you're like, let's hold off on that?
I mean, anything from the KMR data has all been vetted through the USGS, and they're very comfortable with that.
Okay.
Anything from that publication. And what I can do is I can also try and find links. There was, it was written up in a few newspapers. And so that kind of offers a little bit more of a public focused, I guess, perspective on that.
Mm-hmm. Nice.
Yeah, that would be great. Thank you.
That would be awesome. This is so great. I'm so excited. So thank you so much, Stephanie, for all of your hard work. Now